Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you very much for everyone who joining us now for this IBD webinar on the biotechnology, science, business, and society. Today is our 31st seminar with a very special guest. We have today Dr. JAPARENG LALUNG. He's Associate Professor, School of Industrial Technology, University of Science, Malaysia. When I read his CV and his background is very interesting because he grown up in a village, and then he grew up in Sabah, which is the East Malaysia, one of the richest place in the world for biodiversity. And Dr. Lalung, he studied environmental microbiology basically, and he studied in the University of Malaya. University of Malaya, if some of you outside Malaysia don't know, it's the #1 university in Malaysia and among the top 100 university in the world. And he go in different type of study, and we have also studied in between in Oxford University. And he got his PhD in environmental microbiology from one of the leading university in the world, which is Leeds University in UK, and making a significant milestone in his academic journey through different study related to environmental microbiology. He is currently Associate Professor at the School of Industrial Technology, University of Science Malaysia in Penang, where he's continue his inspiring and education for next generation of learners, and he's doing a lot of research related to environmental microbiology. With this, I would like to welcome today our speaker, who are looking too much to hear his very interesting lecture about making hay while the sun shines in Antarctica. Welcome, I think all of us now welcoming our guest speaker of the day. Thank you very much.
Thank you from us very much, Prof. Hisham. Also, thank you very much for the very nice introduction. And thank you very much to the faculty for inviting me to give a speech today. Before I start, can I ask if my slides is appearing now? Can you guys see my slide?
Yes.
Oh, thank you very much. Yes. Thank you very much. So, yes, it has been a great pleasure to be part of your seminar series. And as I heard just now, this is the 31st seminar, so I think it has been going very well. Congratulations for that. I'm excited to share my story, and I hope we all are gonna learn together. I think I have about 40 minutes to talk, and if I talk too long, then please just stop me. And if you have any questions, you can also ask me. So I'm going to start with how was I invited to this seminar series. I actually met Dr. Hasmaliana when we went together to India to Biocon Academy to learn about biotechnology, and we then started being friends. And then when she heard about my research, she said, maybe you can share with us your research. And I was actually very happy. I said, oh, of course, absolutely, we can learn together, hopefully. So the topic that we pick today is about Antarctic research, because I am part of the Malaysian Antarctic research program, and I hope for today we're going to share our Antarctic research experience. I guess making haze means like taking opportunity while the sun shines is actually an English proverb. It means like take the opportunity when you have. So I'm going to talk about the opportunity that I took during my trip to Antarctica. So basically, I had a few trips to Antarctica. The first one was in 2015 and came back in 2016, and the most recent one I actually came back last month. Since February until May last month. And this is how I get the bird support. In Malaysia we have our Antarctic Research Foundation, which is called Yayasan Penyelidikan Antartika Sultan Mizan. So if you have time, you can actually apply for the research grant and birth support to go to Antarctica. They have a few schemes. These schemes opens every year, and it's quite competitive, but I think we can try, and who knows, you might also be getting the chance to go there. Before I start, there are a lot of people to thank, and this one is 52 from the previous trip, and I think there are so many to mention, but they're all in here, mostly from Malaysia and also the British Antarctic Survey. And this is about myself, but I think I'm going to skip this. A bit of my background. I also used to work before, and my research interest is actually water, cyanobacteria, and microalgae, or the microbiology. And for my previous trip, I wrote a book. This was published last year, and I'm writing a book now. I was planning to publish it like in a few months' time. Hopefully, I can do that. This is some of our experience when we were there. And I'm also a coordinator of RC Penang, Regional Center for Expertise Educational for Sustainable Development. It's actually under the United Nations University in Japan. I used to work with, we are currently the consultant for Air Selangor and Next Generation Oil. And this is our research team. Now we have 4 PhD students from Indonesia, Libya, and Malaysia, and we have 3 MSc from China, and 2 students under my supervision. One is from China, Zhang, and Jacobi is from Japan. But he's from Japan, but he's a student at University of Science Malaysia, and this is our workforce team. This is our publication last year, and University of Science Malaysia, in case if you don't know, it's actually a university in the northern part of Malaysia. We're quite good with global impact ranking, which is sustainable development goal. We were ranked 4th last year and also in 2022. And the center another center that I work with, Center for Global Sustainability Studies, we're actually in charge of the ranking. And this is the school that I work, environmental technology. And we have masters in environmental science and also masters in sustainable development practice. But for now, okay, I want to talk about how did it start. So basically, I think making haze means taking advantage when you have, and this is what happened. Actually, before I come back from doing my PhD, I've always been inspired by this one professor called Professor Nazalan. He did his PhD at Cornell, and I've been following his work because I think he's a very good professor and very renowned. He's retired now, and one day there was a viva, and he was the chairperson, and I was the examiner. And that was our first encounter, and I thought like maybe I better get to know him so that I can learn from him. And I told him that I was a bit interested in Antarctic research, and he basically led me towards the right direction. And Dr. Faridina was my first collaborator, and we worked together and write the proposal together. And finally, I've got a chance to go to Antarctica. What I'm saying is that normally when we manage to do something, it's not just about us, but I think it's about the people that we're surrounding with. I think it's very important to be surrounded by the right people for the right things that we wanted to do, and hopefully then they will give us support and manage to achieve what we want. So always take advantage when you have a chance to. When we go there, we've got a lot of expectations because my study was about molecular microbiology. I was there to collect samples, so it sounds quite jargon when I talk to people. They think I was like going there for DNA extractions, identifying species, metagenomics, and functional genes. And you can see a lot of things on Facebook because part of the responsibility towards the funding is that we need to publicize the Antarctic research, especially the Malaysian Antarctic research program. So I need to put something on Facebook when I have internet, but actually it was quite hard to get internet connection eventually because we only use satellite phone over there. And then, of course, you hear a lot of says, like people have a lot of questions like, were you having fun? Did you manage to cuddle penguin? Did you see whale, iceberg, polar bear? Or is it cold even, and obviously it's very cold there. And then these are also among the things they say, like, can I see your pictures when you're taking samples? To be honest, it's very difficult because when you're doing work, we basically do it very seriously, and we are always making haste while the sun shines because we always need to be hurry because we don't really have much time. The weather has the final say. So, and we need to collect our samples. So time proposing is actually not much. Unless for this one, this is near the base, so I have a bit of chance to actually snap. And I've got a friend Jim. He was taking the samples when the pictures when I was taking my samples. But most of the time when we do work, we don't have time to pause, and we were rushing with time. So this is the journey. Normally, I start with Malaysia since I've been going like for all my trips. I went with the British Antarctic Survey. So we first go to the UK. We went to the airport in the UK. It's either Bryce Norton, the military airport, or the uh, Heathrow if we're using civilians airplanes. And then from there, we went south to the Falkland Islands or to Punta Arenas in Chile. We make our way and then we go to Antarctica. We can either fly to Antarctica or we can sail to Antarctica and coming back the same way or different ways. So it's quite a long journey, about 50,000 kilometer. And one way, it's probably more than 1 week. So the good thing about doing this Antarctic research is that next time when you drive for 5 or 6 hours, you don't care anymore, or maybe even for 12 hours, because you have experience of like 2 weeks one way. So every other journey seems to be quite simple in the future. So that's kind of nice, you know. And we have a lot of kids, you know, like this is quite big. I think this is more than 20 kilo of our kids alone. And because we can't really do any mistakes when we go there. You have to have the right warm clothes. But luckily, because we're working with the British Antarctic Survey, so it's quite easy because they're very well prepared. They have like more than 100 years experience. I wouldn't go it myself. I wouldn't go there myself, but with having them tag on with them, making life so much easier. And then we go to Falkland Islands. This is my trips from 2015. But last year I didn't go to I didn't go to Falkland Islands. I went to Chile, and we sail. And this year I've got a chance to actually sail with Sir David Attenborough, which is one of the most advanced research vessels in the world. I think this is the second year that the SDA actually went to Antarctica. So I feel very happy. Actually, when I saw this, when I saw they were constructing these new vessels, I was very excited, and that's one of the reasons I decided to go again. And we also fly the Dash 7, and this is the conditions inside. This is how when we're going, and this is how the base look like. And we have some spare for food. Like this is like in the middle of nowhere. In case if the base is being burned, they still have food supply. The ship is quite nice and communications. And there are a lot of rules and regulation in the base, mostly because I haven't updated with my recent trip. So this is from the previous one from a few years ago. They have a lot of food in there, and so basically you're not going to be struggle with your food because there is enough food, and the British has more than 100 years experience, so it should be okay. And health and safety, I think this is very important to know. Before you go to Antarctica, if you want to do this project, you need to be healthy because they check for everything, your teeth, your medical conditions, and they also train you. You have to be a certified personal, what do you call, first aid, and also you need to do personal safety training, which is the course which is needed for seafarers to actually travel by the sea on research vessels. So there are a lot of things that we need to get ourselves prepared with before we go. So instead of course, we need the funding, we need the fitness, we need the knowledge, we need to write a proposal for the science that we wanted to do research on. And because so they're gonna train you on how to fold these crevasses, you know, so these like holes in between snow, ice. So they normally have flags to ensure that you do not go there because otherwise, if you drop, you're gonna be in trouble. And of course, there will be a lot of seals, and they teach you how to get rid of the seals and how to not to get bitten by the seals. And they also have recycling, energy mostly using, this one is solar tube from Sydney Island, and you got this till lamp which is very traditional in case if you get stuck somewhere in the middle of nowhere without electricity. Then we also taught on how to use this one. Parts and the transportation, they also train you on how to use skidoo, and basically everybody is going to be trained, because over there we have to do basically everything. You're the scientist, you're also the one who need to carry things when you have to, and uh, at different times maybe you need to carry other people. So we were trained, these are from two different times when I go. If you look at the schedule, it's actually a different one. So one is from 2016 and the other one is from this year. And sometimes you're also being trained to use all these very, how do I say, like amazing what is this machine. And the one on the left is actually the cockpit of the flight. I didn't get a chance to actually fly a plane this year, but a lot of my, because this was actually just on the hangar, which is the station or the place where they keep the plane. But my friends did become co-pilot, but I didn't get a chance this year yet. But I hope next time if I go, I can actually fly a plane in Antarctica. But that has not happened yet. But I drove that Volvo, which was actually very nice. And we also travel with boat, so we need to be very well equipped. Like you see this, the one below, we have like our warm clothes, and also we have our float, and that actually make movement quite difficult, because sometimes the swell or the wave is very very strong, and you cannot fall into the sea because it's very cold. I can go up to like minus 20, I mean the temperature. So we really need proper health and safety equipment. And then at certain times when we climb, we need certain equipments to I mean certain kind of snow transportation or snow boot to walk, ramp. And of course sometimes people come and visit the base, we look at them from afar. But also I went there for alga collection, and I think this year uh, was quite challenging because I went towards the end of season. The good thing about going towards the end of season is that there are not many scientists anymore. I was the only scientist when we were there at this during my time. But of course, there is also other people who are doing their project or who are assisting other project. But the thing is, when there are not so many scientists, you can make use of the equipments. But also for me, because when I wanted to collect algae, I need to see the soil and the algae. But towards the end of season, there will be a lot of snow, and all the algae is covered by the snow. And when it's all white, don't even know where to dig. So I showed the pictures from my previous trip, which is 2016. When I come during early seasons, I was happy. But this year, it was quite difficult. So I was battling between the weather and my sampling activities. But I think all the time, the more we went, the more we learn about new things. I'm happy, so that at least next time I knew that I should come during early seasons. So I try to collect as many as possible, algae, fungus, and the cyanobacteria and water. The idea is actually to come back and isolate the microbes so that we can see the functional genes. I can talk about the science, but I think it's going to be very complicated because we have multiple projects going on at the same time. We wanted to look at many functional genes that are actually present in Antarctica, and then we wanted to see if we can make use of them. So one of the things that we're working with now with Dani, which is the Indonesian student, and also with Jay, the Japanese student, is on finding some strands that can actually reduce mercury. We already found the genes, and we are going to run the uh, mercury reduction experiments in the lab this year, and hopefully we can get some results. For the gene isolation and identification, we already published the paper, but for the mercury reduction, we hope to publish the paper this year if we can get some good results. And we collect algae even from snow. Those are snow algae. You can see it's a bit reddish, and we also have greenish snow algae because they're growing quite well because of abundance of the sun during summer. And I think if you know, some algae are actually can grow well, even though at nutrient limited conditions, because they can fix the nitrogen from the atmosphere, just like the beans. And some algae can also do the same thing. So these are the kind of algae that I went there and collect. These are algae on snow, and we sampled them and took them home. And we also grow them in the lab while waiting for our time to come back home. And I think it's very important to know when you're doing sampling, the planning is very important. We need to know what attire to use, how much food to bring, who to go with. So when we're doing sampling, because in Antarctica you cannot go alone, you have to work in a team. And for me this year, almost a lot of my samplings are actually using boats. So I need to liaise with the boating team, and at least we need 3 people with the boating team and another people who are going to accompany me to do the sampling. So there will be at least 5 of us at one sampling occasion. And bear in mind, all these people have their own job, and they also have their responsibility. And not everything is about me. So I need to plan in advance and when and what to do. So sampling can be quite a massive thing to plan about because there are a lot of logistics to be involved and people's time, and also the amount of sample that we wanted to get, because some of the places actually under special protected area, Antarctic special protected area. So we need to know where what we can sample and what we cannot. But I can tell you this. We were very lucky because we were with the British Antarctic survey team. I can tell you that they're very, very well experienced, and their support and help is a lot. So although things are not easy, but they make it possible. And for this time, 2023, basically it's quite easy for me because I have different project. This time I determine the sampling points, but for my sampling exercise in 2015 and 16, it was quite difficult because I was a part of the project was actually to follow the sampling sites that somebody else has done before. Professor Brody, he did it in the 70s. So I need to find the sampling location, but it was very difficult because there was no Google map over there. So I couldn't find the sampling locations easily. After 40 years, we have some differences. There were descriptions about sampling points, but I think after 4 years, a lot of changes has happened. And sometimes there is a place that you need to collect algae from, but you realize that you can only see snow, or you get it completely wrong when it's supposed to be land, but you can see it's actually sea. So it was quite tough. And then we go back to basic because there is no Google at that time. You cannot Google. Internet was very slow. So I use a map and then I draw them. But luckily now I think things has changed. In May this year, in April this year, when we left the vessel, Sir David Attenborough, they actually brought the satellite receiver for Starlink, and I think from this year onwards, they are gonna have proper Google over there, and they can use internet. I think that's gonna be a very big game change, and I think there will be internet. So I think with having internet at the station, it's gonna make life so much nicer and so much easier. This year, I do not culture any of the samples in the lab because I learned that I can just do it, whatever that I can do in Malaysia. I'm not going to do it in Antarctica. But previously, I didn't know whether the culture was going to grow or not. So I started doing the experiments even when I was there. But this time, no, because I'm very, very used to it already. Because initially, I was worried if uh, I culture these strains on at temperature above freezing. I do not know whether they're going to grow, but now after so many experience, we realize that yes, we are going to be interested into with the culture that can grow at ambient or room temperature up to like 28, 29. Are there going to be any strains? The answer is yes. The strains that which are going to die? The answer is yes. But what we're interested in is that we're just going to culture it at a ambient temperature in Malaysia, room temperature, and any culture that survived, that's the culture that we're going to be working with because we know that this culture can survive in Antarctica, and now we know that they can survive at temperature about 30, and then the range of their survival is actually quite big, and we wanted to work with them because we can then use them for whatever the purpose that we would need. We can use them in colder temperature, and we can also use them in warmer temperature. So this is one of the things that we do, and we've got a lot of questions about this because they say, can they survive? And will you lose some strength? The answer is yes, they can survive. Will we lose some strength? Yes, we will. But we are only interested about the tough ones. I think there is a say like when the going gets tough, the tough gets going. So we're just going to work with the strengths which is able to tolerate a very, very low temperature and very, very high temperature. And there, there are strengths which like that, and we found them, and then we're working with them currently. Sometimes we have problems when you cannot Google it, the little problem become very difficult, but then you just need to think and uh, try to get to solve a problem like this one because I didn't have bottle that time. This year is nice because the lab is big, but last time I couldn't find a place to culture my strains. I didn't. You don't have the because the battery dish was all gone at this time, because it first it becomes so dry quickly and all my media dried out and I didn't have any media left to do my culture, so I autoclaved them, but then the petri dish couldn't stand it. So but I still need to set the media because that's the all media that I have, and finally I can use this 1.5 ml tube, and because it's transparent I can also grow my culture. So the whole thing that we need to do requires a lot of planning and also a lot of thinking. uh, if there is internet, it's very easy. But during my time when I was about almost 10 years ago, it was difficult because there was no internet. But now, if you go, it's going to be easy because there is internet. This is the post office in Falkland, which we send our sample to Malaysia. So of course, there are many other science in the base. We normally focus into our own experiment. In my case, it's algae, but other people are doing on krill. There are cameras anyway on iceberg. And I wanted to talk about the weather, like, some easy things which normally is very easy to do, like this closing this seal, which should be very easy. But then when you have thick glove and minus 20, this one is going to become a very, very difficult job. And we need to get prepared. Normally, I open all these plastic bags before I go sampling because I know that on the field the wind is going to be very strong, and it's going to be very hard to open them. So basically, during our sampling, the weather has the final say, or in particular, it's the wind and the snow that has the final say. When it's windy or gusty, like up to 50 or 40 knots, you're not going to go out. And we don't really worry about the temperature, even though it's minus 20 or minus 25. It's okay if there is no wind. You really need to plan with tides as well. This is from my trip in Sydney. And of course, we have lots and lots of ice experience. Beautiful. And the bluer the ice, the older it is. This is probably more than 1,000 years, 10,000 years old. and you see snowflakes. And at Sydney, you can see a bit of plants, but in Rotera, the stations that I went visit, there is no plants because it's even further down south. You can see a lot of penguins, and oh, of course, sometimes squirrel attack. If you can see, this is attacking my colleague. Felix was the doctor at the station, and Vicky was the field guide. So we went sampling together, and the squirrel was attacking us, and we know that it's going to attack the upper part, so that's why Vicky was putting a bag on top of her head so that the squirrel will attack the bag, not himself. If you can see that Felix was bending down just to make sure that the squirrel or the bat will not attack his head. And all these things we need to know, and also we were told before we go there so that we can avoid any confrontations with the wildlife. I like to talk about this picture because I think this is the only place in the world that I can see a seal on a runaway. If you see the two lights, it's actually a runaway or the airport. So the seal was like just resting on the runaway of the airport. So I find it very, very interesting. I took the picture. It's a blonde seal. This fur seal, they used to be almost extinct because they were used for, the fur was used for coats, making jackets, and until the population almost disappeared, but now they are back. But the blonde one is still very rare. And we also see other animals, and sometimes this time I'm very lucky because a few times when we went for sampling we see humpback whale, which is very nice to see, very big, very good experience. They're very big, and we see them many times, but that day I was very lucky to actually be able to capture the image. We see many things like this is like reflection of the suns on on the ice in the morning by sunrise. It's actually amazing. And we saw this view from our base, and this is just before snowing, before we went for sampling. But the thing is, normally when the weather is nice, we go for sampling, and very often we miss the magnificent weather. We actually miss because we were like preparing or taking samples, because we always need to take the chance, like when you say making haste when the sun shines, we need to take chance when the weather is good, because it sometimes the weather is just bad for like days and days, even for weeks. So when the weather is good, we that is the time when we plan to go out and take samples. But also normally that is the time when you can actually take picture and enjoy the weather. But at the same times the work has be done, because photography or is actually not the main purpose of going there, and because we went there for sample collection. But I always just take the chance, like whenever I've got a chance to snap, I'll just do it anyway. So this was just before the snow, and I think the morning was absolutely beautiful. And of course, when we were there, when we go there, because the government and the funding actually was a lot, like for this trip this year, I've got two fundings, and the trip from the Falkland Islands into Antarctica and back, that cost £27,500. So it's quite a lot, and I've got three research grants to actually support the research. One is FRGS, and the other one about 160,000, and then I've got another one from the Sultan Mizan Antarctic Foundation, 150,000, and another short-term grant from Malaysia, from University of Science Malaysia, that's 40,000. So basically, the whole thing is almost like about 500,000 ringgit. So when we spend that money, we need to make use and take advantage of everything. So what I do is normally I also write, because you at that time inspiration is actually quite a lot, motivation was quite high. So we also, I try to work and make use of my time when I have the idea to write. So I have written a book. I mean, for the previous trip, the book was published. But for the current one, I'm still preparing for it. And normally in the morning, I will work from 8 o'clock, and until like late, almost 7 before dinner, and try to make use of all the time and the idea that we have. But at the same time, we also have a lot of things to do. Like you can play ski, you can like, because being from Malaysia, we didn't really have much chance to ski, because it's warm, there is no snow. So sometimes you also feel like, okay, when I'm there, should I just work or miss the opportunity to actually ski in Antarctica, or should I also take some break and do the recreational activity? So I decided, for me, I am going to do some recreational activity on weekends, which is on Friday, on Saturday and Sunday. So yes, I did go for skiing a few times, I think twice, and then I also went for ski-doo, and we also went for climbing, and of course a lot of photography whenever I've got a chance to do it. I think to get prepared for to go there, of course, you need fitness and take care of your teeth. This time there is doctor, because this is the main station. But previously there were only five of us, and eight and five at different times. So there was no doctor. But if you go to the main one, which is Trotera, there is a doctor, two doctors there. So that was good. So there is no issue for me this time. And the doctors are very, very nice people. And yeah, you're gonna carry like quite heavy stuff and water most of the time. So fitness is very important. If you're not very fit, I think it's gonna be quite difficult to go there. You can still go, but it might be a bit challenging. And I noticed that everybody who was there, they're actually very fit. Even the doctors, uh, both of them, when we were there, they did marathon. They basically run half. The female doctor run half marathon, and the male doctor run the full marathon on the runaway. When I was there and I took the picture, so it means that even the doctors are very, very fit. Well, they normally are. So fitness is very important to get ready to do Antarctic research. And sometimes we climb and we cannot. If you can't make it, it's going to be quite difficult. Like in this case, my field guide was much stronger than me. I was very weak, and which in a way is good, because he was like very far ahead and he took my picture. He zoomed me from afar, but obviously I looked very weak that time because I was very tired. And I think also I want to talk about this, because I think at first I only have before I go there, I thought I need to understand a lot about molecular DNA part and the specific the aseptic method of sample collection, the media to grow, the growth condition, and all which I know already. So I think to do Antarctic research, to bear in mind that you are going to be there, and at that time, for me, I was alone. There's nobody, almost very little people to discuss with. So go there well prepared, and I know that we wanted to learn new things when we go to a place, but in this case, it's best for you to know as many things as possible, because sometimes if you don't know something quite basic, we're going to waste the whole trip and the whole funding and the whole time to go there. So try to get as much knowledge as possible before we go there. And that's why for British Antarctic Survey, for any scientist, they normally require a senior scientist, because for junior ones, like a student, unless they have a supervisor going together with them, they probably will not be allowed, because it's going to be difficult to make decisions when things matter. Like in my case last time, when I run out of media, when the plates were melting, I really need to step in and think of what can I do to make use of the situation. So like knowledge about how to convert map into GPS form. So these things we really, really need to learn. How to get rid of seal, how to not fall into crevasses. Like another thing also, I think when we go there again, when I said making haste while the sun shines, is actually take advantage of everything, because we're just gonna go there. I always assume that, although I went a few times, but I always assume that it might be the only time that I go there. So I learned like how to take photo, like what is the shutter speed, because time is very limited. And then we need to fully utilize it. And if you want to learn, you can, but then it means that you're gonna make use your expensive time. And like at minus 30 degrees Celsius, opening a sealable plastic bag can be quite a challenge. Ping pong ball situation. What happens if you are right in the middle of ping pong, inside a ping pong ball, which means that you go left, right, up and down. There is nothing. It's all white. What you do is actually you stay until the weather is getting good. So uh, sometimes even for a bit of going out, you need to make plan. This is especially true when I was at Sydney Island, which is my previous trip, and Rothera, which is my current trip. Last month is not so challenging because it's quite a big station and there were a lot of help. Of course, motivation, and then you really need, because it's not easy. There are a lot of logistics you need to arrange, and you need to push yourself. Like that time, I need to see the deputy vice chancellor, because I really need to buy my flight ticket and really need to get a release from the university. So these are the challenge that you just have to bulldoze it. It's not going to be easy, because what I'm saying is that we only see the nice pictures, but the story behind it's almost like just the tip of the iceberg. There are a lot of things that we need to sort out before we go and never give up. And also like working with people, understanding British culture, and this is also very important. Like homesick is not even counted because we are homesick, but then if you don't really have much time to miss your family because you have so many things to do. And we also need to understand about the rules regulation, permit to the UK, permit to Malaysia, where can you post it, where will the ship dock, how to get to the post office if you need the post office, the rules and regulations of the post office, the rules and regulations of British Antarctic Survey, and the rules and regulations of the British Quarantine Department, the permit. So these things we need to know. Otherwise, because if we don't follow the rules, the sample that we have taken that costs us a lot of money, it's going to go into waste if you cannot bring them back to Malaysia. So all this logistics, all this knowledge is actually very important. It doesn't look very serious because when being scientists, what we concern the most is about the science, but in reality, it is just a small portion of it. The real thing that need to be done for Antarctic research is actually far beyond that. Like this permit and licensing is quite a tough thing to do, but I think in Malaysia we're quite lucky because we have the Sultan Mizan Foundation, who is actually going to advise and help you to do this. And we really need to seek for help and seek for information, because otherwise you cannot just do it all by yourself. So one of our challenges was to send it back to Malaysia. Like now, all my samples are still on the ship because we sail from Antarctica to the Falkland Islands, and I fly from the Falkland Islands to England and England to Malaysia. But my samples are still now on the Sir David Attenborough. They are now sailing from Falkland Islands to Cambridge, and I'm going to go back to the UK where the British Antarctic Survey is located at Cambridge, and I'm going to collect a sample from Cambridge and bring them back to Malaysia, hopefully in August or September this year. So these are the final product. Out of everything that I went there for collection, those are for this year. Those are the only one box that I sent. It's about 30 cm times 30 cm times 30 cm in size, about 33.6 kilo. It contains my culture, the samples that I took from Rothera this year, and it is still in a box, frozen in the Sir David Attenborough vessel, and it's going to arrive hopefully in Cambridge, hopefully in June this year. Oh, June, which is now, yeah. I think I'm going to email and ask them if it has arrived, and then I'm going to make a plan to actually come and collect it and work with it when I reach back Malaysia. And managing perception is also very important, and because there will be like a lot of recommendations, especially people from Malaysia. Why not this? Why not that? Why don't you post like this? Why don't you post like that? And why don't you pakai baju Melayu and show everyone? So it's gonna be quite difficult to listen to whatever people say. At the end of the day, I think it's very important for you to know that you just do what's best for you, because you know it well. And you know, when people when you take a lot of pictures, people are going to say like, oh, he's actually just having fun. There is no picture of him taking samples. But in reality, when you take samples, you cannot take picture. Of course, when we were taking picture on the skidoo, yes, when we were posing, because when you really riding the skidoo, nobody's going to take your picture. You cannot even take picture because it's dangerous. So this perception is actually also sometimes you need to manage, because there's a lot of says that Antarctic research is actually very expensive, and what's the benefit towards the society. And yeah, these are some of the challenges that we need to overcome, which is managing perception. And I think it has been about 47 minutes. So I'm gonna conclude with also how can you join the Antarctic research research in Malaysia. We have Yayasan Penyelidikan Antartika Sultan Mizan. They release fundings, and they also support a birth trip to Antarctica. And we have National Antarctic Research Council in Malaysia, and also we have Malaysian Antarctic Research Program. So talk Google this and see what they're doing. And if you want uh, to know more, I think the information is available in their website, especially YPASM, uh, which is in Jalan Sultan Ismail, next to it's basically the same office as the Academy Science Malaysia. And we have SPARK partnership. I think these are the universities that are actually taking part in the Yayasan Penyelidikan Antartika Sultan Mizan SPARK partnership, when all the members of these universities can actually go to Antarctica and apply for the funding and the trip to Antarctica. And there is a bad support. Yeah, but I think I want to give also a tips if any students wants to do this. And of course, you cannot do study yourself. You know, to if you're not do research in Antarctica, you cannot just go yourself. You need to have a partner, because it's almost impossible to just go there by yourself. Uh, Normally, I prefer to work with British Antarctic Survey, because they are very, very particular about health and safety. I've never worked with any other institution, so I do not know. But I know that the British are very, very concerned about safety. They basically do not tolerate any accident. And uh, going to do research in Antarctica is tough, and you cannot do mistake, because otherwise you will die. So I work with them, but bear in mind that to collaborate with them, uh, they also need us to contribute our knowledge. You know, so we probably also need to speak to them and convince them that we can actually be a good partner, because expecting them to do everything for us is not going to be possible. You know, like just like if you just go like, I don't know, what am I going to do, and can you tell me what to do? It's not going to happen like that. So tell them that what you want to do, and hopefully they can match you with their expertise. Okay. Yeah, this is what I mean. They will not infiltrate you into their system if you're not beneficial to them at all. This is life, you know. Yeah, it's very important to know that they will only take you if you're going to be useful to them. It's very hard to get free things in life. So put this in mind. I think being a student, what you can do is actually to learn and make yourself qualified. And then once you are, then you have something to offer for yourself. Thank you very much. I hope that's okay for now.
Thank you very much, Dr. Jeb. Wow, that was wonderful and very eye-opening sharing. Yeah, I hope the audience also enjoys what Dr. Jeb has shared with us. Okay, so with the also Dr. Jeb has shared the very amazing photos. The photos are from you, right? You capture them
All of
yourself.
them from me, yes.
Okay,
Yes.
so yeah, because I know that Dr. Jo is actually a photographer as well, for for the information, for your information. Okay, so how long normally you the trip to the Antarctic?
Okay. Thank you very much, Dr. Asmaliana. And again, thanks to you for inviting. It's nice to see you again, old friends.
Yes.
Yeah, we are. Okay, so for this time, I went there in March, the most recent one, because depending on, to be honest, like before I go this year, I always make haste while the sun shines. I always kill a few birds with one stone. So this time, I asked the British Antarctic Survey. I told them that, okay, let's be honest. I like the science. I wanted to do the science, but also I'm going to enjoy myself. So
Yeah.
I told them that, yeah, I heard about the ship, the new ship, Sir David Attenborough. I
see.
want to be in that ship. And
Uh-huh.
I asked them, can I go there by flying and come back by the ship? So that I have
Mm.
no experience of flying and come back by the ship. And they say,
Okay.
yes. So for that, we look at the time frame that I need. I need about 6 weeks times of sampling. And so we look at the possibility of fitting me into both method of transportation, which is going by flying and coming back by
Mm.
the boat. So I went
see.
there in February and come back, reach back Malaysia in May. So the
Wow.
whole, yeah,
It's quite
yeah,
long, yeah, the trip.
it's quite, yeah, yeah.
Uh-huh.
Yeah, but what happened is that Dr. Hasmalini, I think we both are lecturers. What happened is that when we go somewhere for 3 months, our class is still going on. So
Yeah,
for
yeah,
me,
yeah. How do
yeah,
you manage that?
like now I'm in a big,
Trouble.
huge, yes, because I push all my classes towards the end,
I
and then
see.
now I have like so many lectures. But then you either do it or not. Like, yeah,
Yeah, yeah,
otherwise,
yeah.
yeah. So yes,
I can imagine that. So,
yeah.
so it's lucky, yeah, because your management is supporting you.
Yes,
Yeah.
very much. Yeah, yeah.
So
We
normally,
we, we mhm.
how many people from your team that go together?
One.
Only you.
Yes.
I
Yeah.
see.
Yeah. In
Okay,
fact,
okay.
from Malaysia, for the British Antarctic Survey, this time is only one.
Mm-hmm.
Mhm.
Okay, okay. So I think we have some question from the audience. Okay, from Mr. Muhammad Amirul. Good evening, doctor. When you reach there, what can you see the condition of Antarctica due to climate change? Is that because you have been there 2 times, right? When in 2016 and in 2023. So can you see any changes of the environment due to this climate change effect?
Thank you very much, Muhammad Amirul. And the answer is no. Because if I said yes, then I'm going to be bluffing. Because when we talk about climate change, we're talking about Antarctica. Antarctica is big and vast continent.
Mm-hmm.
It's maybe twice the size of Australia. It's very big.
Mm-hmm.
Uh, it's not small. So, and then when I went there, I never go to the same place, so I cannot see the difference between one place and another. And
Mm-hmm.
also, I go there at different time of the. I think if you come in winter, it's going to be ice. If you're going to come in summer, the ice is going
Mm-hmm.
to be melting. So, like going there for me, I cannot tell the difference because A, I go to different place. B, I I went to a different timing.
I
So
see.
even when I was there, I can still see, like at the beginning when I come and the ice was melting, but then when I stay until approaching winter, the ice is going to be start freezing. But
Mm-hmm.
I think in order to know whether climate change is happening or not, you need to go at exactly the same place at exactly the same time, then you can compare. But I can
Mm-hmm.
tell you, we have a lot of glacier, and I asked around, yes, the glacier has been melting. And this is from what I heard people say, or from what our conversation with my colleagues or my climate scientists. But from my own eyes, no, the answer is I couldn't tell.
Mm. Okay.
Is that answer?
So not yeah, yeah, yeah. Because you've been to different places, so you cannot say you see in the changes, yeah, right? Okay.
Mhm.
And normally for sampling, the scientists never normally they go during the winter, the summertime. Am I right? Only
Yes,
only during the summertime.
depending on your research. For my research, because I
Mm-hmm.
wanted to find the algae grows when there is sunlight.
Uh,
During
I
summertime, it's
see.
completely dark, they're gonna die. So yeah, so I
Okay.
come in summer. Yeah.
I see. Okay. Okay. Do we have any question from the audience? Okay, from Miss Ida Madiha. Hello, doctor. Thank you so much for your sharing. My question is, how do you identify the exact sampling location? Is it needed to be identified before the trip? Thank you, doctor.
Oh, thank you very much, Ida. Very good question. Thank you very much. Yeah. Actually, I would say depending on the purpose of the research. For my previous trip in 2015 and 16, I need to identify the sampling location because of the nature of the study. So basically, for that time, I need to see decadal changes, like the difference between the the algae diversity in that place 40 years ago when the sampling was done by Professor Peter Brody from Canterbury, New Zealand. And then I repeated the same experiment in 2015 and 16, and we wanted to see the differences. That time, we really need to find exact location so that we can compare. And it was tough because I only have map. And I have GPS. We have GPS location and map. But 40 years ago, GPS was not as accurate as it was now. So uh, I need to use the map. I look at the map that he used and I basically estimate it to as close as I could. So basically, it is not going to be exactly the same to all of them. So, but I basically estimate and look at the map and draw it manually. Yes,
Okay,
and
I see.
that was very tough. Yeah, and there's no Mr. Google, and
Mm.
yeah, it was very tough. But for my recent trip in May, no, because the nature of the study was different. For me, I was then the first person who tried to find the algae there, so I determined the sampling locations. So I just see,
Oh,
yeah, so I just see where I found the algae, and it's gonna be like that, and pick up the GPS easy. Yeah, so it
Okay.
depends on the.
Yeah, yeah, yeah. That was quite challenging, because you are the only researcher that uh, had to find the algae. So it's quite
Uh,
challenging.
I mean, no. How do I say? Yes, you're right. But I wasn't the only one who need to find the algae. During the time when I was there, I was the only one. But there are other people
Mm,
who come
I see.
and go. Yeah.
Okay.
But during my time, I was over there. I was the only
Mm.
one. Yeah.
You mentioned in your talk just now, there's a lab inside. Is it in the ship?
There is a lab in the ship. There is a lab at the station. Bear in mind,
I
the
see.
ship cost probably billion ringgits. They have culture lab, which is far more advanced than our lab here. I can tell you, they have the money. They have culture lab. I tell you, the equipments of the British Antarctic Survey. To me, one of the
Uh-huh.
reasons I wanted to join the British Antarctic Survey because
Mhm.
is that I get access to billions of worth of equipments. It's not cheap.
Mhm.
So and this is like how do I say? This is high end and cutting edge technology that they're having. I can tell you what, even when I was there, we have the capacity of using PCR, which is designed for
Uh-huh.
COVID-19 testing. So
Oh,
they even have special PCR machine which is optimized with kits for COVID-19 testing.
I
And then
see.
yeah, and then being scientist, I think you can read the manual and you understand. Although you're never
I-
being trained, but you understand the primer, and then I can,
Mhm.
yeah. They're very good.
Okay, meaning that the lab there is quite, complete.
Yes,
I see.
yes.
I thought it's like very limited, limited. Okay. So
In all
other
sense, they're very
uh,
advanced.
very well equipped. So other than British Antarctic Survey, is there any other institutes? There must be other institutes that fund this research to Antarctic, right? The trip to Antarctic.
Okay, first I think in Antarctica is a big continent, so we have
Mm-hmm.
British base, China base, American base, Ukraine base, Russia base, Japan base, and many.
Mm-hmm.
But we work together with British Antarctic Survey. So
Mm-hmm.
uh, that since we have collaboration with British Antarctic Survey, only then we are allowed to enter their station because
I
this
see.
is their property, and expensive. And
Uh-huh. Uh-huh.
once you enter, the good thing is that once you have collaborations, you are equal. You
I
are
see.
treated as well as any other scientist or any other British
Mm-hmm.
citizens in that area. So once you
Mm-hmm.
enter, you're equal. There is no Malaysian,
Mm-hmm.
no British. They treat
Mm-hmm.
you as if you're one of them. So
Mm-hmm.
and we are lucky because universities, because Malaysia has collaboration with the UK. In
Mm.
order to, no, nobody's going to allow us to enter their station because we're going to do all their secrets.
Yes.
We're going to
Yes.
make use of their sophisticated machine. Like
Mm-hmm.
like I can go and say like I need to go sampling tomorrow, so I need to use the boat, and they will
Yes.
provide all the boat driver, the people, the
Oh.
supporting team, even the field guide, even doctors to go and go with you. This is not
Mm.
easy. This is not cheap.
Mm.
So we need collaborations. Otherwise, they wouldn't do it. So
Mm-hmm.
I prefer to work with British Antarctic Survey because I know this collaboration because Malaysia has collaboration with them. Um, uh, but how do I say? So for us, uh, for the funding to go here, there are many things that we need to find money for. A
Yes.
is the consumable or the equipments that you need to do when
Mm.
you come and do the research in Malaysia, because you can just go take samples there, but then are you going to run the experiments? How do you pay the PhD students? So I
Mm-hmm.
applied for grants with Yayasan Penyelidikan Antartika Sultan.
Mm-hmm.
So actually, the first thing I did was I applied for FRGS using similar project, but using tropical strains. The
Mm-hmm.
idea is the same, but tropical strains. And once it's almost successful with tropical strains, I then change the game and try to use Antarctic strains. And
Mm-hmm.
I apply for another grant with Yayasan Penyelidikan Antartika Sultan Mizan. And
Mm-hmm.
once I got the grant, then I apply for a trip to go there because I know that I
Okay.
need backup to do the
Yeah, yeah.
experiments first. Yeah, and then
Mm-hmm.
I apply to go to Antarctica. And but the whole thing is that when you join the British team, they basically allow you to go there, although the worth of the, they call it the birth support, is 27,500 British pounds, or almost about 150,000 ringgit Malaysia. But
Mm-hmm.
you also need some money to buy for your plane ticket from Malaysia to Punta Arenas. So I still need that money. Then I apply for another short-term grant from University Science Malaysia. So basically, throw grants together. Yeah, actually, the grant management is very important.
Mm, yeah, yeah.
One by one.
It's not easy. It looks very easy to go there, but actually not. It's not.
No, you appeal so many times in it take years of planning. No.
Okay, do you have any other question from the audience? Okay, from Miss Lilina. Good afternoon, Dr. Japareng. What is your biggest discovery from the journey?
Wow. This is a very good question. The answer first is I do not know what is the biggest discovery that I found from this journey. Uh, I don't know, but I'm gonna figure out something. What I think is best. I think uh, I can learn about life a lot. When I go there, one thing I really learned is that it's a mind. I think it's very important among us, from developing country or from Malaysia, especially when we have collaboration with the UK. We always expect them to teach us, give us money, and give us everything. But I tell you what, that was long time ago. Now we are climbing up. I think it's very important for us to understand that in order for you to collaborate with somebody, regardless to whoever they are, you need to contribute something. So remember that to be part of the team, they're not just going to take anyone. If you have nothing to contribute, nobody's going to contribute back to you. That is the harsh reality of life. So what I'm saying is that whenever being a student, whenever you have a chance to actually improvise yourself, learn the skill when you are a student, so that you can be beneficial to others, so that in the future, if you do collaborations, you can get something from them, and you can also give something to them. Because if you just tag, it's not going to be sustainable. I don't know if that makes sense. So that to me is a big take, and sometimes you don't realize that um, it's very important to study, to learn when you can. And that's why I said make haste when the sun shines. Whenever you have a chance to learn. And also very basic knowledge that you need, like learn how to swim. If you cannot swim,
Mm,
you
mm,
cannot go. Take care of your teeth. If you have rotten
mm,
teeth, they're not going to allow you to go. And stay
mm.
fit. If not, you cannot go. So a lot of things is actually like on tip of the iceberg. You can see the ice, but beneath, then well
Yes.
prepare yourself to be a good person. So that
Mm-hmm, mm-hmm.
yeah, so that easy for
Yeah,
you to do things.
yeah, you talk about your current research just now about the strain that can reduce the mercury level. So that is your current research, I guess?
No, that was the
No.
previous one. The
Okay,
current
okay.
research is about. I tell you what, the current
Uh,
research is actually. I go to Antarctica. If I can tell you to find strains that
mm,
can break plastic.
I see. Yeah,
Yeah,
yeah. That
because we
I
know.
indeed
Mhm.
that was the the current situation, yeah, in the microplastic pollution, right?
Yes,
Mm,
yes, 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 yes. So basically, my proposal, I have to like make a. I need to make a solid proposal on this because we think that the strains in Antarctica is actually quite strong, and
mm.
then the pollution is very little. So in order for them to survive, they basically need to make use of any nutrients or elements that they can, and we hope that plastic will be one of them.
Oh,
So yeah,
I thought my microorganism from Antarctic is quite, not strong because they they live in very low temperature. Because I have experience working with the Antarctic protein, and it's very very, tough to make them soluble, even to make them soluble.
I I I call the kill the diva technique. I don't know if that makes sense. What I do is that I'm gonna because if you have one strains, then it's very difficult. You only have that one option. But for me, I'm just gonna culture as many as possible, and the one who can survive, that's the one that I'm gonna work with. The rest is
Okay,
gonna
okay,
die. There will be a lot of
yeah.
them. Yeah, so I said like all the diva is gonna die. The survivor will.
Okay. Okay, maybe our last question from the audience. Our final question, maybe. Okay, from Miss Riani. Hi, Doctor. From your experience, what are the challenges working with Antarctic microorganism in your research?
Oh, thank you, Sureni. Again, very good questions. Okay, the challenge is actually, to be honest, is sample collection. That's the biggest
Mm,
challenge because unlike any other science that we do in Malaysia, you can easily get the sample. But for Antarctic research, the biggest chunk or the biggest effort to actually do the research is go and get the sample. So that is the biggest challenge. But once you get back the sample in Malaysia, suppose the challenge is actually to grow them in uh, ambient temperature, which is like 29 high, like above freezing, like even 20, 25. But I already have my skill the diva technique. So I already just grow them and then whoever doesn't grow, I'm not going to work with them because I'm not going to like lower the temperature of the lab. No, some will grow. So I'm just going to work with the strains that can work with ambient temperature. I treat them equally as our tropical strains, and there
okay.
are lots of
I
them.
see. I
Yeah,
see. I think
lots
you
of
can.
them.
I think you can patent the scan the diva technique. Okay. I think so. I think that's the final question that we have from the audience. So we are also running out of time now. So thank you very much, Dr. Jeb, for very very wonderful sharing your experience, your trip to Antarctic, and also your research in biotechnology related to the Antarctic. Okay, so in behalf of Institute of Bioproduct Development, I would like to express our sincerest gratitude for you for accepting our invitation to our webinar series today, and for spending your precious time, although you have so many class to catch up. So I hope good luck to you to catch your students later. Okay, for our audience, thank you very much for joining us today. So I hope you can benefit and have some uh, uh, take home message from what Dr. Japareng has shared with us today. So with that, I end our webinar today with Assalamualaikum warahmatullahi wabarakatuh, and good day. Have a good day. So take care, everyone. Bye. See you.